Welcome back. Today we're talking about fixatives according to composition. So we are grouping fixatives or classifying fixatives based on what they are made of. Simple fixatives. So simple fixatives are fixatives that consist of a single active chemical agent. So these fixatives are either used alone or as components in compound fixatives. So simple fixatives can be further categorized based on their mode of action. For example, we have coagulant fixatives, which precipitate proteins and disrupt cellular structures to preserve tissue. Examples of coagulant fixatives, we have alcohols like ethanol and methanol that are commonly used in cytology, mercury chloride, which is a strong coagulant, picric acid, which coagulates proteins and preserves glycogen, and acetone, commonly used for rapid fixation and enzyme. Second category under simple fixative is the non-coagulant fixatives, which form crosslinks in proteins preserving cellular structure. For example, formaldehyde, which is the commonly used fixative in histology that penetrates tissue well and preserves its morphology. We have glutaraldehyde that provides excellent preservation for electron microscopy, and osmium tetroxide that is commonly used to fix lipids and as well used in electron microscopy. Second classification is compound fixatives. So compound fixatives are mixtures of two or more chemical agents. For example, formalin or the 10% neutral buffered formalin and Bowen solution. So the compound fixatives can further be categorized into microanatomical fixatives and cytological fixatives. Microanatomical fixatives are fixatives that permit the general microscopic study of tissue structures. In other words, they are used to preserve the overall structure and architecture of tissues. Classical example is use of 10% neutral buffered formalin and use of Bowen's solution, Zenker's solution, formal sublimate, and others. So the second category under compound fixatives are the cytological fixatives. So cytological fixatives preserve specific parts and particular microscopic elements of the cell. And these cytological fixatives can either be nuclear fixatives that preserve the nuclear structures like chromosomes. Example of nuclear fixatives, we have Fleming fluid, Caronized fluid, Bowen's fluid, Newcomer's fluid, Hayde-Sousa, and others. The second category under cytological fixative are the cytoplasmic fixatives. So cytoplasmic fixatives preserve cytoplasmic structures. Examples is Kelly's fluid, Rigard's fluid, or Orthi's fluid. The third category is histochemical fixatives. Histochemical fixatives preserve specific chemical constituents in tissues for subsequent analysis. For example, called acetone for enzyme studies and alcoholic fixatives for glycogen and other carbohydrates. Additionally, we have major groups of fixatives. One, we have aldehyde, oxidizing agents, alcohol-based fixatives, and metallic groups of fixatives. So aldehyde fixatives contain aldehyde groups. So examples of aldehyde fixatives, we have formaldehyde, glutaraldehyde, and glycol. So these aldehydes work by cross-linking proteins. They form covalent bonds, especially with amino groups in proteins, to stabilize and preserve the structure of tissues. The second group are oxidizing agents. Oxidizing agents, these are chemicals that oxidize or remove electrons from biomolecules. Oxidizing agents act by oxidizing biomolecules such as proteins and carbohydrates, leading to structural stabilization. They break specific bonds, for example the disulfide bonds, and form new stable linkages. So these fixatives are particularly useful for preserving lipids and fine structural details in tissues. The third group, we have alcoholic fixatives, which majorly contain alcohols like ethanol or methanol. These fixatives act by dehydrating tissues. So the dehydration causes proteins to precipitate, effectively locking them in place. And these alcoholic fixatives are rapid and effective. However, they usually cause shrinkage or tissue distortion. The last group is metallic fixatives, which contain metal salts. So metallic fixatives work by binding metal ions to tissue components, which stabilizes their structure. Examples, we have mercury or chromium salts that precipitate proteins or carbohydrates to preserve tissue structure. Thank you for listening.